Well, welcome back to our series of videos on the topic of cell division. Now, in the last video, we looked at what happens during meiosis I, right? And we saw that during meiosis I, pairs of homologous chromosomes separate and end up in different daughter cells, so that each daughter cell ends up with half the number of chromosomes. Now, in this video, we're going to take a look at what happens during meiosis II. So in meiosis II, what happens is that the pairs of sister chromatids will separate. So you take a look at this daughter cell here. It has half the number of chromosomes, but the chromosomes are still replicated. They still are a pair of sister chromatids. So this pair of sister chromatids needs to separate so that we can finish our meiosis. So here's where we stop, right? That meiosis I, the big idea is that pairs of homologous chromosomes separate. And now we're going to enter into meiosis II. Now the good thing is that meiosis II is a lot easier than meiosis I, because meiosis II is actually very similar to mitosis. In fact, it's actually identical to mitosis, except that you're dealing with half the number of chromosomes. So at the end of meiosis I, we had two daughter cells. And each daughter cell contains half the number of chromosomes. So in this case, each daughter cell only has two chromosomes remaining. And the two chromosomes are still replicated, right? That's why they look X-shaped. So what's going to happen in meiosis II is that these pair of sister chromatids are going to be separated from one another. So in order to prepare for the next division, we have prophase II. And in prophase II, right, the centrioles move to opposite poles within each daughter cell. The nuclear envelope in each daughter cell breaks down, and the spindle starts to form again to prepare for the next division. And in metaphase II, right, what we see then is that the pairs of sister chromatids will line up along the equator. Right, so this is very similar to metaphase in mitosis, except with half the number of chromosomes. So we're getting ready to separate the pairs of sister chromatids. So the sister chromatids are attached to spindle fibers from opposite poles, right? So in this pair of sister chromatids here, one chromatid is attached to spindle fibers from this pole, and the other chromatid will be attached to spindle fibers from the other pole. And then we have anaphase II, right? In anaphase II, the centromeres of each pair of sister chromatids will divide, and the sister chromatids will then be pulled to opposite poles as the spindle fibers shorten. So again, this is very similar to anaphase in mitosis, right? The pairs of sister chromatids will separate and be pulled to opposite poles. And after the sister chromatids separate, each of them can be known as a daughter chromosome. Then finally, we have telophase II and cytokinesis. So you can see that in telophase II, right, the daughter chromosomes, right, the sister chromatids have moved to opposite poles. Right, the nuclear envelope has reformed, the spindle has broken down, and then there will be a cleavage furrow formed, right? And each of the daughter cells will be divided into two. So we'll end up with 4 daughter cells. 4 haploid daughter cells are formed. Haploid because they only have 2 chromosomes each. So in summary, here's what happens in meiosis. Right, we start with a parent cell with a diploid number of chromosomes. So in this example, that's 4. Right, the chromosomes will replicate themselves. So here in this cell, we end up with 4 replicated chromosomes. Then during meiosis I, the pairs of homologous chromosomes will separate. And each daughter cell will end up with 2 chromosomes, but the 2 chromosomes are replicated. So each daughter cell will have 2 replicated chromosomes. And then in meiosis II, the pairs of sister chromatids will separate. So we end up with 4 haploid daughter cells. Right? And each of these will go on to be a gamete, a sperm or an egg. And that's it really for meiosis. So it can be a bit complex, but let's remind ourselves of what the objectives were. We need to be familiar with what happens during each stage of meiosis. And very often in exams, they'll test you by giving you diagrams of cells and ask you which one is in which stage. Is it meiosis I, or is it meiosis II, or is it mitosis? And you need to be able to tell which picture shows what phase. Right, so with that, I'll end here. And in our last video, we're going to be taking a look at how meiosis produces variation. So how does meiosis produce different combinations of traits, different combinations of alleles in a population of organisms?